Okay, any questions from last time? Anything on chromatin structure? We're gonna talk about. Okay. Yeah, so you guys ready? So there was a question on hats and HDACs. Those are part of the chromatin remodeling complexes. So the hats are the ones that add acetyl groups to histones. So the connection you want to make is that the histones, when modified like that with an acetyl group, that tends to open them up a little bit more. And then that allows the movement of those histones around the DNA easier. And then the HDACs are antagonistic. So they're the ones that take off the acetyl groups. And then that allows the histones to be more closely packed and to hold the DNA tightly. So the remodeling complexes, I would assume, would use both those types of activities to modify the histone, the nucleosomes, the histones in the nucleosomes, right? And be able to then remodel or shuffle those around. So that's, I mean, I'm not gonna actually, I guess you should associate what hat activity leads to. So that's what I would try to do is think, okay, hats tend to loosen the chromatin, and that's going to open up the DNA, and so HDACs would be the opposite, right? That's kind of what I would do. Thank you. Oh, and by the way, congrats to the women's basketball team. I didn't know what were the results from last night. Did they put you someplace?
Yeah,
Notre Dame. Oh, when is that? Friday. Wow, exciting. Okay, I expect you to be in. No, I was kidding. So, well, good luck, and rest, practice hard this one. We know you'll give it your best. Okay, good. So, other questions, other things we got to talk about. So we got that. So I'm going to give you, so remember what that is. That's an example of epigenetic control, right? So above DNA sequence, we see chromatin has an effect on transcriptional initiation. And you have to deal with that, right? I'm going to give you one more example of epigenetic kind of regulation, which is kind of interesting. So we do have one more paper this week. So it's not too bad looking at transcription factors and how they are interacting to start transcription from a particular promoter. So it shouldn't be too bad, I think. You guys have covered a lot of the ideas like hybrid proteins and how to use those. And then our exam is next week, right? So try to get through RNA modifications and RNA interference this week. We'll see how it goes. And then we'll be ready for our test next week. And there's no lab tomorrow as well, right? I'll be busy with science fair pretty much all morning. So I won't see you. Unfortunately, it kind of sucks. We got to the cool part where we finally have our plasmids. I think I saw colonies on pretty much every plate from last week. So we finally got our plasmid, and then we don't do anything with it for 3 weeks. So it kind of sucks. So, but I'll be kind of working on that in the background. I'll probably have to take those plasmids and introduce them in the yeast so that you guys are then ready to run your experiments. So you kind of want to keep that in the back of your mind of what we're doing with that project and what was the goal up to this point. You know what we have done. Okay, so that's probably a good review for the test as well. Go back and review what have we been doing in lab and why, and what's been kind of the, not the intricate details of the methods, but what are the techniques that we have been doing, right? So, anything else? Okay, so let's take a look at methylation. So this is kind of a really interesting example of epigenetics. So again, we're looking at effects on the DNA, not really the sequence, because obviously changing the sequence can change transcription. But these effects change the regulation of the transcription of these genes. So methylation, here we're really just talking about literally adding methyl groups, typically to cytosines. That's one of the more common ones, but there are other bases that can be modified as well. But if you think about this, you're adding, that's a pretty bulky group, isn't it? A methyl group. You think about the chemical environment that we've been talking about in DNA. This could change that chemical environment in 3D, which we can see is going to change how proteins interact with it. And we see this used commonly in development. So in mice, they looked at these globin genes, and this is true for humans as well. But in utero, as well as at the very early stages of development, we see one particular globin gene that's active. And then as we go through development, we slowly see the methylation of the promoter regions of this gene, which shut that off. And we actually switch to a different active form of the globin gene. Of course, the globin genes are involved in oxygen carrying, right? In hemoglobin. So we see a clear developmental pattern with some of these genes that are turned off at some stages. And this is never turned back on in that individual. It's never turned back on, at least normally. It's not turned back on. So the methylation here is actually affecting which genes are expressed and when. So it's in development. So it's kind of cool. So the idea here is, right, we've been talking about the access of these regulatory proteins to the promoter region, whether that's the general or specific or specific transcription factors. Now what we can see is that sometimes if we get methylation patterns in here, this could either sometimes block the access that those transcription factors have for the gene, or sometimes they can actually recruit other proteins. So this could even be, we could talk about those chromatin remodeling complexes can actually be recruited through this as well. So it kind of depends on the gene. I don't know if one is more common than the other, but we see examples of both. We see methylation of DNA that can inhibit the binding. So again, that's going to be simply a steric hindrance, right? You can put something in the way and the protein's not able to bind. And if it's a transcription factor, then you're not going to see transcription probably from that. But we also see some what they call CPG islands. So there's a lot of CG stretches that are methylated for one reason or another. And these can actually attract proteins. So you think you could attract maybe a chromatin remodeling complex or even a transcription factor by itself. They could be attracted to this. And then again, this could have some sort of downstream effect on the transcription of that gene, whatever it is. So it's kind of interesting, right? So what do you think? Can DNA, if it is methylated this way, do you think that could possibly be inherited and maybe direct downstream developmental processes from this? So can it be influenced by methylation? It's kind of interesting. And yeah, I think you guys have the answer, right? This is going to be called genetic imprinting. So I don't know if you've ever heard of imprinting. Sometimes they talk about imprinting in terms of animals, right? You can imprint yourself on a baby bird or something, and then it's not able to develop properly. That's not what I'm talking about here. This genetic imprinting is where we're going to actually see DNA that's modified, and that's going to actually allow the offspring to differentiate, if you will, to differentiate between parental chromosomes. So it can actually tell the difference between, or at least tell the difference of which chromosome was inherited from which parent. So it's basically modified DNA that, I guess, how we want to say it, will be treated differently in the offspring. And we're talking about transcription here. So it is going to affect the transcription of these chromosomes in the offspring. This is going to be genetic imprinting. And here's the idea. The idea is maybe you have methylation patterns on the parental DNA. What you're going to imagine is we have proteins again, so like the HATs and the HDACs. We have methylases, methylation proteins that are going to change the methylation patterns as you go through the replication process. So this picture, I just took it from a review, but their idea was that, well, just because you have a parental methylation pattern doesn't mean it's going to be identical in the offspring. In fact, there's been some examples where some offspring, they just basically erase all methylation patterns.
So you start over with kind of a blank slate in the offspring. And that's kind of what they found in mice. So this is a really interesting example in mice. So what they found is that, of course, you have male and female, right? We don't have to talk about, do we have to talk about sex in this class? Hopefully not. How you make baby mice? Okay, good. If you don't know, call your parents, okay? But there's eggs and sperm, right? That's kind of how it happens in animals. In the female, interestingly enough, in the female, they erase a lot of the methylation patterns in the female oocyte, right? So in egg development, that's the female imprint. And then the male imprint, the males actually put a lot of methylation patterns throughout their sperm. So think about the genomes of these eggs and sperm, okay? So they're establishing their own imprints, their own genetic imprints. And this is going to have an effect on which genes are expressed in the offspring. Because if you think about it, right, you get one egg, one sperm, right, from each parent. We each get half our genomes from each parent. So you can imagine then the baby mice are going to be kind of heterozygous in a sense, aren't they? Heterozygous in the sense of methylation patterns now. So we don't know where they are in DNA sequence, right? But in methylation pattern, kind of head. Wow, this is interesting. Okay. And that, you can already imagine, that can have a different effect on the expression of whatever genes, because we're talking mainly about your methylating control regions, right? So promoters or enhancers, something like that. So it's going to affect it at the transcriptional level. So this just kind of shows you can get different patterns from these changes in imprinting patterns, okay? So here's another view of it again. So in the eggs, you kind of erase the methylation patterns. And in the males, they call it de novo. It's kind of from scratch, basically. So they basically methylate all sorts of different genes, okay? So I'm trying to set the stage here, because what we find then is we find some genes that have what's called a differential methylated region, a DMR. So the DMR, depending on if you have female or if you're coming from a mom or a dad, right? They're going to be methylated differently. And what I want you to try to convince you is then that's going to actually affect which genes are expressed, okay? Here's a really cool example. So this enhancer region actually controls two different genes. There's an H19 gene and an IGF. We're just going to focus on the IGF. It's a growth hormone, okay? Depending on the methylation state of that enhancer, that then dictates which of those genes are expressed. Isn't that kind of interesting? One promoter essentially is controlling two separate genes, but only one of those genes is expressed at a time from this particular locus, right? Okay. If there's no, so which one is it? If there's no methylation pattern, we get to see the transcription of the H19 gene. And if there is methylation, then we see the expression of the IGF. So we're not going to talk about the transcription factors, but basically, whichever factor binds here, and one only binds when it's methylated, that's going to dictate which gene is expressed. So far, so good? So, trying to convince you who can methylate things. That methylation can affect the binding of different proteins. The methylation patterns are changed in each parent. Now we're going to see the effect on the offspring. You kind of see where we're going? So the offspring can actually express genes either from maternal or paternal chromosomes. This is the payoff. This is the payoff here. Really interesting. How are we doing? Okay. So this is kind of cool. So what do these guys do? As good scientists, we say, well, how do we study this, right? Well, you start knocking stuff out, right? That's what we're good at. We're good at modifying DNA. I remember the whole beginning of this class, talk about modifying DNA. So what do we do as good scientists? Well, we make a homozygous knockout to a homozygous wild type. That's what we do, looking at the IGF gene. That's the IGF gene. Okay, so what happens in mom? What happens to methylation patterns in mom? Go back to this. And this This is really only true for mice. We have not seen this exactly in humans, but we think this kind of gives a basis for the function of methylation. So we use this to try to understand humans. So what happens again in mice? It's erased in females and imprinted on males, the same loci, right? So with the female, we have a knockout. Well, that's not good, right? What would we expect from a knockout IGF gene? Would we expect a functional protein? It's a minus, right? Minus means mutation. Mutation is generally not good, right? So we have a knocked out mom's gene and we have a dad's gene there, except in the dad's gene, what happens to the IGF allele? In dads, we methylate it, don't we? Not in moms, but in dads. Hey, and that expresses that IGF, right? So, hey, that's pretty good, isn't it? We actually have a functional gene in dad, and that's the one that's expressed according to the methylation pattern. And so this just essentially is a recessive mutation. We don't see it. We would expect with normal heterozygous organisms, we would expect to see the wild type phenotype, wouldn't we? Because we have one good copy, and that's what we learned in genetics from our genetics professor. As long as we have one good copy, we're good, right? Well, not true so much with gene dosage, right? I guess we already disproved that a little bit. So wild type looking baby mice. Okay, what about the, does anybody know what this kind of cross is called? Crisscross? No, it's not a test cross. Close. It's the other one I teach in genetics. Teach test cross, and what did not Watson, I was gonna say Watson, Mendel, when he did his crosses, he tried it from sperm from one flower, and then he took sperm from the other flower. What's that called? Starts with an R. Ends with an L. I'm just picking on it. Reciprocal. It's a reciprocal cross. So what do? We should see the same effect in the reciprocal cross. Oh. It's like, no. So what's going on? Isn't this interesting? We get dwarf baby mice. What? That's not following genetics rules, is it? This is a heterozygous offspring, but what's the problem? The allele is turned off. Yeah, over here, dad's allele is working and it's expressed. Over here, we got the working allele from mom, but in mom, that allele imprint is erased, which means what happens to the IGF gene? It's turned off. What? Wait, we've got a perfectly good gene that's being turned off. And now we have the turned off allele and the other allele, which is a knockout. So now we have a double knockout, basically. Look at that. What do you think? Crazy? So, although we inherited a functional allele, the offspring actually turns it off. It treats it differently based on the methylation pattern. You're probably going, what on earth? Why would we even do anything? Seems so dumb to turn off a good allele, doesn't it? But this has a lot of role in development. You see these methylation patterns do it. So do you guys understand this? You see that one gene is being turned off, not based on its whatever, its coding sequence, but based on its methylation patterns. Listen.
So like in the first example, when you mutated the mother's phenotype, it's not methylated right, so it'll express it, and then that causes the
Right.
second example, mothers we don't change, but it still doesn't express it because there's another methylation. The mutated the father's there, it doesn't methylate either, so it's smaller.
Yeah. So the basic idea is that in both the maternal chromosome, we never express the maternal loci. Come on, guys, flex your muscles though. But in males, it's the male gene that's always expressed, right? Yeah. They call this selfish DNA, right? So there's another theory. Okay, and I don't want to be sexist, maybe it's better to try to express a gene from either the male or the female, right? Maybe there's a developmental advantage here. That's a question mark still, okay? But the mother's gene is never expressed. So they call this kind of a selfish DNA hypothesis, where one parent, you're expressing more of the alleles from one parent because they're the cooler one, right? Maybe. That hasn't been proven. Okay, so what? We don't care about mice. Is there an example in humans? Yeah, so I put that down here. Maybe males have a desire to silence females. Okay, don't quote me on that one. That'll get me in a lot of trouble. Okay, but maybe there's a developmental thing there. Is there an example in humans? Yes. We see lots of examples in humans, including that IGF gene in humans that is affected by methylation patterns. And let me just give you one example of a really interesting syndrome based on this. Has anybody heard of either Engelman's or Prader-Willi? It's a really interesting one. It's about a mutation of 1 million bases. It's a huge mutation. Oh, I forget which chromosome it's on. Did I write it down? Chromosome 15. Chromosome 15. It's 1 million bases, so it's, well, relatively small compared to the rest of the chromosome. But what it does is it actually deletes this area where there's a couple of linked genes that are really important in development. But the reason why it has two names, before they even sequenced the DNA and figured out where the mutation was, they actually described these two syndromes separately from each other. And it was only afterwards that they found out that it was associated with the same mutation on the same chromosome, except it depended on whether you inherited the working chromosome from your mom or from your dad. And that then dictated which syndrome you got. The syndromes are pretty similar. There's pretty significant developmental delays, but there's some phenotypic differences. Like one, I think they're really skinny people. The others are kind of more chubby. So you see, that's why they thought it was two different syndromes at first, because they didn't really match. But we see examples of that in genetics as well, where you get a mutation which actually can give you different phenotypes. I forget what the term for that is, though. But you guys can look it up. So this is kind of interesting. Let me just show you what it looks like. So here's the region at the top, and you have the blue genes and the red gene, and they're controlled again by a DMR, differentially methylated region. So just think of it as part of the promoter. So how does it work in, let's see, I think it's, let me make sure I get this right. I think it's in females. Females is the blue one. Female, yeah, the blue ones are expressed in from the female chromosome. The red ones expressed from the male chromosome. So what happens if you delete, maybe I had it the other way around, but if you delete like the paternal chromosome here, and you look at this, we see that the blue ones are going to be silenced and the red one is expressed. So this one's actually expressed from the, yeah, it's expressed from the maternal. I had it right. Okay. Expressed, yep.
So that's like what the arrow means, like it's expressed.
Yeah. The arrow means transcription, right? That was on the quiz. When you see the arrow, that's where you start transcription. But if you delete it from the maternal chromosome, the red one is silenced and the blue ones are expressed. Oh, okay. So again, we see you don't have to memorize this. I'm trying to show you as an interesting example. So just remember, the red and the blue are controlled by methylation. You either get one or the other. And so it depends on which chromosome you inherited it from, which genes are expressed. Because normally the blue ones are expressed from one chromosome and the red ones expressed from the other. But if you delete it, then the methylation patterns screw everything up. Isn't that interesting? Again, we see we have perfectly good genes here on the mom's chromosome, or the chromosome you got from mom, right? But they're not being expressed because of the methylation pattern. And then, does that give you an advantage? Certainly not. You have a developmentally kind of disabled person from that, even though you have perfectly good genes on there to express. And then the same thing over here. You might be able to express these blue ones from the dads, but not the red one from the moms as you're supposed to. So look them up. It's kind of interesting. So that is Prader-Willi and Engelmann. And it depends which syndrome you get, depends on which chromosome is deleted and which chromosome you inherit the good chromosome. Which parent, sorry, you inherit the good chromosome. So what do you think? Kind of interesting? So again, these are examples of epigenetics, things that don't necessarily affect the DNA sequence if you just ignore the deletion. We're not changing the sequence really, but we're changing these methylation patterns on the DNA, which then affect how they're expressed in the offspring. It's another thing you can blame your parents for. Besides giving you bad genes, they give you bad transcriptionally regulated genes. Is that what you? They probably will look at you like, what else are you going to blame me for? Okay. That's okay. When we hit 30 or so, we all think, oh, yeah, we had pretty good parents after all, didn't we? At least that's what they say. Anyway. I hope so, because none of my teenagers like me right now, so that's okay. I'm like the worst parent ever. That's okay. So questions? Questions here. So anything on chromatin or methylation? Really just two examples of how we see transcription being affected by this.
Maybe I missed something.
That's okay. Which
Well,
slide are you looking at?
The last one, or the one after this.
Yeah. Oh, this one or this one?
The what we present with, yeah. So like you mentioned how this is epigenetics, the distinct methylation is.
Yeah.
But this one, they are the leading like, and so.
On one chromosome, right?
Yeah, so how is that considered? Epigenetics?
Well, just look up here at the top. So, and I stumbled on myself a little bit, but it's the blue ones that are silenced in the female and the red ones silenced in the male. And this is in humans. So when you have wild type copies of them both, it's fine. But if you delete one, so like if you delete the paternal, where you're supposed to express the blue ones now, blue ones are all gone. We have blue ones over there, but they're all silenced. So that's what I'm trying to point out, is that the methylation pattern it seems like it's really rigid. It won't change just because you are missing a good allele of the other one. It always stays the same. So it's always going to be silenced still, even though you have good copies of that gene. And so really what's interesting is here is that it's almost as if the offspring can differentiate between the paternal chromosome and the maternal chromosome when it expresses its genes.
Even though they look the same.
Yeah. And so to me, it's kind of fascinating because, you know, when you're first taught genetics, you're always, oh, you got two copies, they're both expressed, blah, blah, blah. But as we see now, it's so much more complicated than that. Even though you have heterozygous individuals, if we go back to those mice, even though you have these are identical genotypes, aren't they? And so in genetics, we're taught they should be identical phenotypes. They're not. Because there's a whole nother layer of complexity. So that's kind of what I'm trying to show you. You don't have to memorize these genes. You don't have to memorize the methylation patterns in mice. But I want you to just go, wow, we can affect the expression of it without even affecting the sequence. And then what is the developmental role for that in the offspring? So one idea is maybe it's better to express these genes off one chromosome and not the other for whatever reason. There's still a lot of studying got to do.
So as the offspring develops and makes new cells, the DNA is replicated. Does it hit the methylation and add a 5-methyl cytosine, or does it add a cytosine and then methylate it like after? Like I'm confused on that.
Well, no, the cytosines are there. The idea is that the replication machinery will have these methylases associated with them. And so as you're going through replication, right, if you think about meiosis, then in a female mouse, they'll actually demethylate all the histone, or it's not histone, sorry, all these cytosines. So they're going to go along and just chop off all the methyl groups as it's replicated. So then the oocyte genomes will all be blank in terms of methylation patterns. And then in the male, the methylases will just go through and methylate them all. So then, I mean, that's simple. Just think about
Well, we either remove them all or put them all on there, but that's not always the case. That's just mice. And so we see other genes where we have different patterns of methylation. So now you can maybe have a subset of genes that you're going to methylate in females. That's girl power. We want to express those genes from the mom because they're superior, right? I'll stop boasting about males, right? Is that kind of something? So you can actually mark subsets of genes to be expressed from female chromosomes, right? There's so many things we could imagine that could occur here. No, I'm not convincing you. I get it. Now you guys got to step back and think about it. These are one of the thought things where you're not going to get super complex test questions on this. This is where you need to step back and go, how does this affect transcription? That's really what this whole discussion has been about, is transcription initiation regulation. Right?
Whether the offspring is male or female, will they treat the same methylation pattern differently?
Oh, right. So that's probably another layer on that. At least with these genes, I think they treat them the same, but there's probably other genes where they are recognized differently in male and female offspring. But I have to go back and check for examples. Yeah, it's good. That's probably another layer. Yeah.
Isn't this example very close to like in the metaboard of rat book to say that?
Yeah, right. And so each gene probably can be treated differently. This gene, though, are not. The methylation pattern is not recognized differently in male and female offspring. This, I think, is what he was asking. So these offspring, they would recognize this the same, but there's probably other genes that are different. Okay, cool. So roll that around in your brain. Probably keep you up at night or something. Okay, and we're gonna be done. We're gonna be done with this one here. So that is lecture, whatever. Let's see, what was it? 14. So 14 is chromatin. You want to think about how chromatin and how methylation patterns, and there are other examples, right? I gave you a list. There's non-coding RNAs. All these things can have an effect on the initiation of different genes. Okay, so they just add another layer of complexity to the initiation process. So you put that together with all the other transcription factor stuff I'm talking about. Right? So again, you do want to list out, I could ask what kind of things affect transcription initiation. So the main thing you're going to talk about are what? Transcription factors, right? Having the right general transcription factors there, as well as maybe the correct specific transcription factors, and then interacting together. And then when you talk about chromatin and methylation, those just basically affect the ability of those factors to bind, right? So that's where you really want to focus in on. How do transcription factors work? I think there's a question on the quiz about how they have specific parts to them that make them work, right? I'll be grading those quizzes this week, so I'll give you feedback, hopefully in the next day or two, okay? Yeah, how do they work? And then, of course, the interplay. So allosteric, do they have to have the right structure? What happens if we change the structure? That kind of stuff. Right? When somebody shoot me an email and tell me to put up the study guide, maybe I'll add some information to my really short study guides. Okay, I'm going to do that before I commit. Don't put too many swear words in there. Okay, so questions on any of that? Then chromatin, methylation, anything? I want to start in on RNA. Now we're going to look at post-transcriptional things. Okay, so really what we've been talking about are pre-transcription. Now we're in 15, right? So it's gonna work. So we've been talking all about transcriptional control. So even before we get the RNA, a lot of steps in there for both prokaryotes and eukaryotes. Now we want to look at processing. Just because you get an RNA doesn't mean you're going to get protein from it. That's one mistake I made as a grad student, making that assumption. Because there's a lot of stuff. Not only is there processing, but there's also a lot of localization. So where are the RNAs? Where sometimes they can be masked. And they can be silenced until a certain point, and then they can be basically unmasked and allowed to be translated. So it's a lot of stuff that can go along with this. So here we talked about the transcriptional regulation, we talked about chromatin, we talked about regulatory events at the promoter region. All of these things are going to come together to affect the transcription for that. After transcription, so post-transcription, we're going to see, wow, now there's lots of variations in the processing. And we can have proteins that can silence the RNA. We can have RNA interference. So we're going to see a lot of non-coding RNAs that can affect the expression of mRNAs. So great, another layer of complexity when it comes to the expression of genes. And then not listed on here, but it's also splicing. So we're going to see the splicing process actually allows us to get more than one type of RNA from one gene. So this is really going to increase the diversity of proteins that we can get to that. And then even then, do you think we're going to get translation from that? Maybe, maybe not. So, and I would add RNA editing. We probably won't get the RNA editing today, but there's another layer where you can continue to change the RNAs even after we transcribe them. Okay, so you guys love this class, don't you? Oh man, it's like Gen Bio and steroids. Nobody told me about all the stuff we didn't talk about in Gen Bio. This is why I love it, folks. There's just so much complexity here. It's really cool to see it. So back to some of our differences. We talked about the different RNA polymerases and how they could see different genes. We talked a lot about transcription factors, so hopefully you're feeling more comfortable with them. If not, come and talk to me, right? We can go through it, talk about the different transcription factors. What are some of the changes we see? We see now there's actually going to be modifications, and this actually occurs during transcription. And this is only for eukaryotes, right? Now we're really just talking about eukaryotes. Prokaryotes, I don't think there are any post-transcriptional modifications. You just start translating that thing right away. But in eukaryotes, we're going to see these modifications like the 5' cap, the poly-A tail. These are going to be necessary for stability and translation. Can be necessary for the ribosome to identify these. So without them, we don't see translation. Do you have a question? And then with the splicing process, this is crazy. We're actually going to take out the introns and splice together the exons, which you can't really do that in a haphazard manner. You have to make sure it's all correct and in the right order and done without mutation. Okay, so these are super important things. So here's typical modifications. We're going to learn that the GCAP is necessary for the ribosome to recognize the RNA. So when we talk about translation, I'll mention that the ribosome binds here and actually starts scanning down the RNA to look for the start codon, but that'll be after our exam. And then you have the coding region. Remember that the coding region is made up of all those introns and exons, and we have a start and stop codons. So realize transcription usually starts ahead of that coding region. So sometimes the people forget that. And then the transcription continues into the UTR. So in the UTR, we see these other signals that are going to be necessary for splicing. We see a poly-A tail. There's literally a poly-A polymerase that adds something like 250 A nucleotides to the end. So long tail, which is associated with stability. That tail will actually degrade over time. And if it gets too short, the whole RNA becomes unstable and it's degraded. So RNAs have a half-life, if you want to think about it. They're not around all the time. So these things are going to be necessary for stability. And then the coding region with their introns and exons, we're going to see that in general, the exons are going to be spliced or glued together to make the mature RNA. It's kind of what we're talking about. So we start with a pre-mRNA and we go to a mature mRNA. And we have to splice out the introns, which are then destroyed typically to get our functional RNA with our modifications. Oh, yes.
Is the poly degradation, is that just something that happens in solution, or is that a different sub?
Yeah, there are nucleases which will chop them off at a certain rate. So I think the cell kind of does that on purpose, if you want to think about it that way. But not all RNAs are degraded at the same rate. So I think there's other things that influence the rate of degradation of the RNAs. So some RNAs are around for a super short amount of time. Some are longer. So there has to be other mechanisms as well. What happens if you lose a poly A tail? Rate of gene synthesis goes way down. Yeah. What happens if you lose both the cap and the poly A tail? Protein synthesis way down versus having them both. Okay. So they are super important. They're also going to be important for transport. So I'll mention them also that the most transcription occurs where? In a eukaryote? Nucleus. Most translation occurs in the either cytoplasm or at the ER. So they have to be transported out of the nucleus typically, and then hooked up with a ribosome. So these modifications are also important for that. A lot of RNAs, if they're not modified correctly, they don't make it out of the nucleus. So they're not going to be translated. What about splicing? You think we can just cut these things wherever they are and just put them together randomly and say, hey, we have functional protein? What do you think? Some of my questions are really dumb, aren't they? Do I have a timer on this? Not really. And this is kind of crazy. I mean, do you have to make sure that the reading frame, you guys know what a reading frame is, don't you? I haven't really talked about it, but the reading frame are the codons in the exons. Do they need to be in the right order? Can't just skip a nucleotide here or there and say it's fine.
I just like to say they keep putting a baby on you.
Yeah. Well, you don't like the name. It just depends on what mood I'm in when I'm making these little quizzes. If it's more of a snarky mood, then yeah, I'll put stuff in there. So if you don't remember the reading frame, that's where the ribosome is going to read it in 3 nucleotides of 3. So those are the codons. So yes, you have to make sure that when you're splicing these things together, you guys say yes? One person? One person says maybe. It's not really done. When you're putting these exons together, we're going to see this is done by the spliceosome. What a cool name is that? Remember, OME is a huge complex of proteins and RNAs in this case. The spliceosome is going to bind to our pre-mRNAs. And again, they're going to recognize sequences on the RNA, do that. And then they're going to clip out the intron through a really kind of complicated process. But then when you put these exons together, you better make sure that's in the correct reading frame. Because even if you miss just one nucleotide, then you're going to get frame shifts. We did talk about frame shifts, right? Way back when. So the spliceosome is a super important job to make sure it does this correctly. And then on top of that, we're going to see it doesn't always splice things always in the same pattern. You can actually get different splicing patterns. Yes.
How does a spliceosome or other regulatory protein to recognize the sequence or whatever it may be that makes the introns different than the exons? So how's it different?
Yeah. As it turns out, so you're talking about these junctions. I mean, what is an intron versus an exon, really, is what you're asking. And so there, we're going to see there is evidence for sequences, conserved sequences that the spliceosome will recognize, but they're not always the same either. So let me see if I answer that in the next couple of slides here, okay? One thing that you want to recognize, though, is looking at these RNA binding proteins, they bind in a specific manner. So these binding proteins are going to be able to recognize the different parts of the RNA. This is the spliceosome and the splicing process in yeast and C. elegans, because I've looked at a few of these genes in C. elegans and nematodes. But they bind in a specific manner, and in yeast, this has been really thoroughly elucidated. So they've spent a lot of time looking at this. But they bind in a certain manner, in a certain way, and in order to be able to splice these out and degrade the intron, any of you cowboys or cowgirls in here? You know what a lariat is? Really? I usually always have one kind of like horse person in here. Now, a lariat is the rope. It's the rope that they use to catch whatever. Nobody wanted to fess up to it, I guess. So they make a lariat structure with the intron, cut it out, and then usually the lariat is degraded. So it's cut back into pieces. So it seems kind of wasteful, right? To make an RNA and then cut chunks of it out and degrade it. I will give you a hint. I don't know if it's gonna be this week or next week. Sometimes though, we have RNA, non-coding RNAs in these introns. So they're not always just junk. They're not always just junk, okay? But that's kind of the process, or the simplified process, where they cut out these introns. And what we're gonna see is there are conserved sequences on either side where they can recognize this. They actually showed this in yeast, where they looked for sequences that were required. These were actually signal sequences that kind of signaled the spliceosome and to show where the splicing should occur. So what would happen, you know, if you delete it, do you see any splicing? You delete the signal that's required. If you put the signal in front of a different intron, what do you get? You get splicing to that intron. Put the signal somewhere else, you get splicing to that intron, or sorry, that exon. So they saw sequences that were required for this. And then they also saw sequences that were required for these things to bind to. So you notice that a few of these bind to the exon intron junctions. Some of them bind to exons. And then so the whole spliceosome basically is going to work together to do this process. But I wanted to show you this. Have you guys ever seen histograms like this? So what this says is that the bigger the letter, the more conserved the sequence that they see in these organisms. So this was kind of a cool paper that they did in bioinformatics. Wow, 20 years ago. So what they looked for is they looked for splice acceptor sites and splice donor sites. So this would be a splice donor site over here and a splice acceptor. So you're going to bring those two exons together. So they looked for sequences that were conserved. And guess what? Did they find some? This is a Arabidopsis, C. elegans, Drosophila, Oryzae is a rice. Look at that. So you start to see sequences that are highly conserved between species that basically dictate where the splice is going to start and where the splice is going to go to. And hey, is the start codon also conserved? Yeah, right. AUG is almost always the start codon.
So when you're trying to splice out RNA that's not necessarily introns or it's an exon that you don't want to show it's a different gene or to have different expression.
Right.
Is that a spliceosome that does that, or is that something else that can cut out that for your RNA?
Let me just let's skip this one. We'll come back to it. Let me just give you a, what do they call it? A cliffhanger for Wednesday. So in addition to those splice sites, we're going to see there are sometimes strong signals and weak consensus signals. These can then also dictate which exons are going to be included. I think that's what you're asking.
Yeah.
So it's not all the time every exon that's included, but some of them can be skipped. So we're going to start right there, is that in addition to binding to those splice sites, they can also dictate which splice sites you're going to use based on these signals. Is that okay? And I don't know if I really answered your question yet, but it's these conserved sequences that are going to actually dictate what is an exon from what is an intron. Because they always look different, don't they? The exons and introns, they don't look the same. So there has to be some conserved sequences that I can't think of the right word, that determine what is a splice site and what isn't a splice site. Maybe we'll just start right there. Okay, have a great day. Hopefully they got you excited for science today. Happy Monday. Stay safe out there. It is Monday the 13th, right? So just be wary of crosswalks and stuff like that. Garfield's worst day today.
That's what it is.
It's okay. How goes?
On